back to it a lot today. So yes, just our agenda. Yesterday, we covered an introduction to Ensemble. We looked at how we can visualize genomic regions in the browser, and we looked at the gene annotation that is available in Ensemble bacteria. Unfortunately, we didn't have time to start the Ensemble variant effect predictor, so we're going to start that today. And we're going to continue on and talk about the comparative genomics data we've got available. So these include gene trees, alignments, and homologs. And I'm going to show you how you can visualize your own data in Ensemble. So how you can upload your own files to visualize it and map it against a reference genome. And then at the end of the course, we'll have a little summary. I'll give you some time for some feedback. So the feedback is always useful to us to improve our courses for future participants. And I'll also show you a few resources where you can find more information about training in Ensemble and in bioinformatics in general. Okay, so let's begin with the Ensemble variant effect predictor. So what is VEP? VEP is a tool, VEP is variant effect predictor. I'm just going to call it VEP from now on because it makes it a bit easier to say. So VEP is a tool that you can use to annotate and predict the functional consequences of your own variants. So let's say you just received your sequencing data, you've called your variants from the data, and you found a variant or a mutation in the genome. So what you can do is you can run that mutation through VEP and find out what the consequence of that mutation is on the gene. And I'm saying mutation because we use, well, when I was still working in microbes, we used the word mutation for bacteria, but in Ensemble, these are variants. So whenever you see this word variant, that's just referring to a mutation in the genome. But how does Ensemble VEP actually predict these variant consequences? So what we do is we take each variant and we plot them along the genome, and we predict the functional consequence of the variant depending on where it falls within a gene. So on my slide, what you're seeing is just a diagram of a gene, and these boxes represent exonic regions in the gene, and these lines represent intronic regions in the gene. Anything that's filled, like this orange box here, is a coding exon, and then anything that is empty, like this box here, is a non-coding exon. So if we were to take a variant, for example, and we find that the variant falls outside of the gene, so let's say the 5' or the 3' region, what we infer is that it's unlikely to affect the gene itself, but it may affect the transcription. If the variant falls within the coding region of a gene, so these orange boxes here, the variant can have two effects. So it can have a synonymous or a non-synonymous effect. And if a variant has a synonymous effect, the protein sequence remains the same, or in other words, there's no change in the amino acid. And if the variant has a non-synonymous effect, there is a change in the amino acid sequence, and therefore a change in the protein sequence. So this means that the variant is likely to have a much greater consequence to the gene and the protein function itself. We also call non-synonymous variants missense variants. And we have this really useful diagram in the Ensemble website in the documentation to show you, to just visually depict where, what consequence we apply depending on where the variant falls in the gene. So yesterday I mentioned that while most bacteria don't undergo splicing, there has been evidence that alternative splicing does occur in bacteria, although this is rare. So for most of the bacteria species you see in Ensemble, these variants would only fall within exonic regions or upstream or downstream regions. Okay, so to summarize, in Ensemble, we take the variant that you run through VET, we plot the variant against the genome, and depending on where it falls within the gene, we apply a consequence to the variant. So similar to gene functions, which have special terms called gene ontology or GO terms, variant consequences also have special terms. And we call these sequence ontology or SO terms. And it's just a standardized way of describing the consequence of a variant. And I don't expect you to read this table at all, but what I want to show you here is that each sequence ontology term has its kind of own color, and you don't need to memorize this color coding because it will always be available to you in the documentation. Kindly increase your volume. Okay, you'd like me to talk louder? Is that right?
I can hear you okay, Louise. So I just want to double check it's not a technical issue.
Sorry?
I can hear you okay.
Okay, let me just switch my microphone. Maybe that'll help.
Okay.
Test, test.
That's loud and clear for me.
Okay, wonderful.
Oh.
Wonderful. Okay, great. Cool. So back to the sequence ontology table. So these sequence ontology terms have their own kind of color coding, and you don't have to memorize any of this because you can always find this in the documentation in Ensemble. And there is a URL at the bottom of my slide on here. And these sequence ontology terms as well, you can rank them by impact, so how much it would affect a gene. But please just note that this is a subjective ranking, and just because a variant has an impact of high doesn't necessarily mean that the variant has a disruptive effect on the gene. Okay, so what can you do with VEP, or how can you use VEP? So first of all, what you need to do is you need to input your data into one of these variant formats. For bacteria, these first two formats will be the most relevant. Then your data is processed by VEP, and it searches Ensemble and any external databases to give you information such as the effect of the variant on the gene, the allele frequency, if available, although this is very rare, and a list of publications. So just a preview of these different input formats that you can use. The Ensemble default VEP format looks like this. So this is a tab-separated file. Each column represents a type of information for each variant, and each row represents one variant. So this first example in this Ensemble default format is a variant on chromosome 1. This is the start coordinate. This is the end coordinate. Then we've got the reference allele followed by the alternative allele. Sometimes you can, if you want, you can also add additional information. So you can tell VEP what strand your variant is located on. So 1 is on the forward strand and -1 is on the reverse strand. And if you want, you can give each of your variants an ID. But these two options are these two columns are optional, so you don't have to supply VEP with them. The variant calling format, VCF, is similar. So again, a tab-separated file, and each line represents one variant. So this first variant here is located on chromosome 1. This is the position, the coordinate. We've got an ID, so we've just called this variant var1. The reference allele is A, alternative allele is C, And then when you do your variant calling, often you would have some additional information as well. But these columns are optional. So you don't need to supply VEP with these columns. There are 3 different interfaces for VEP. So you can use the web interface on the Ensembl Bacteria browser. You can do more custom variant annotation by using the command line version, or you can even integrate VEP into your own pipeline via the REST API. Today, I will show you how to use the web interface. Okay, so we're going to move on to a demonstration now. So we've identified the following deletion in Staphylococcus aureus in the N315 genome. And what we're going to do is we're going to use VEP to find out what genes this variant affects and what the most severe consequence is. So we have our variant in the Ensembl default VEP format. So we've got the chromosome, in our case, because Staphylococcus only has 1 chromosome, we just call this chromosome. Then we've got the start position, end position, and then our allele, our reference allele, and then the alternative allele. In our case, a deletion, so there's just a hyphen in here. If you'd like to follow along on your screen, I will copy and paste the species and the variant into the chat. And what I'm going to do is I'm going to switch to my browser and open up a new tab and go to bacteria.ensembl.org. Okay, so we're on the homepage of Ensembl Bacteria. And what we want to do is we want to go to this link that says tools at the top of the page. And then you can open up the variant effect predictor in the table. So the first entry in the table. Okay, so this is the input form for the variant effect predictor. Whenever you see a question mark like this, what you can do is you can open up a help page for this. This will open it in a new tab in case you've forgotten how to use it, or if you wanted more information about how the tool works. In our case, I'll leave it at that, but please feel free to explore the help page in your own time during the exercise. So the first thing we want to do is we want to check our species of interest in the first box. So at the moment, we've got E. coli selected in here. This is not our species of interest, so I'm going to delete that from the list and click on Change Species. And the species we're interested in is Staphylococcus aureus. And I have the GCA ID copied. So I'm just going to enter it in here, which makes it easier to find rather than, alternatively, you could also find your species using these drop-down menus, but we know which one we want to work with, and we have the GCA ID, so I'm just going to search for that instead. Okay, so we've got Staphylococcus aureus, subspecies aureus, N315, and that's indeed the one I am interested in. So I'm going to add that to my list. And if you scroll to the top of the menu, just make sure that you've added your species on the right-hand side here. I'm going to close the menu. And I'm back on the input form for VEP. So next, you want to name your job. It says it's optional, but I highly recommend that you put a name, a descriptive name for your VEP job here, because in a few weeks' time, you won't remember what analysis you've done in this job. So please use something descriptive. I am going to call this S aureus deletion. Okay, then we've got the input data. So you can either paste your data into the text box. You can upload your file up to 50 MB in size. So whenever you see an underline like this, you can always hover over it with your mouse to open up a description. Or you can provide a URL if your file is bigger than 50 MB in size. We have our variant copied, so I'm going to just paste it into the text box. Next, we've got some additional configurations. So here you can tell VEP if you want to get any additional information about the variant. So under identifiers, you can get the protein ID that the variant affects, any UniProt IDs if there are any present, HGVS notation. We also have variant synonyms, any co-located variants. So this one says, this one means, do you want VEP to tell you if the variant already exists? So has someone else identified this mutation in bacteria? If so, yes. So by default, we always show you this, but you can also turn this off. Then we've got additional annotations, so information about the protein, about the transcript, and so on. Any predictions? So in this case, we've got phenotype data and citation, phenotype data and gene ontology information and conservation scores. And then you also have filtering options, so you can filter your results before you even see your results, before you even see your final results. We don't have this turned on by default because in case you might miss out on any interesting variants. You can always apply filters after you've seen your results. And then we've got some advanced options as well. So I've kept everything in the default as a default setting. So I'm going to run my job by clicking this green button here at the bottom. Oh. Okay. So it said here, oh, sorry, just show you. It says here the input format is invalid or not recognized. So what you can do in this case is I'm just going to check everything to see if that's right, because it did work just earlier today. My gene species, I'm going to check if it's the correct species. Yep. Hmm. Okay, odd. So the format is correct. So just as a reminder, the first column is the chromosome. In our case, because it's a bacterium and it only has one chromosome, we just call this chromosome. Then we've got the start position, end position, our reference allele, and then the reverse, the alternative allele. In our case, it's a deletion, so it's just a hyphen. When I click on run, for some reason, VEP does not recognize it. But when I do an instant run, so if you ever have a list of variants and you just want to quickly look at the results of one particular variant, you can use this option to instantly run VEP for one variant. And as you can see on here, it worked. So this is something that that's happening on our end. And what I'm going to do is I'm going to report this to my colleagues and see what's happened there, because it did work yesterday, which is odd. But we have the results anyway, so let's just go through the results for now. So if it didn't work for you, if it said input format not recognized, just click on run. Oh, actually, you know what? It might be the spaces. Welcome to bioinformatics, where spaces mean a lot. Let's run that again. Yeah, that was it.
It's
So
definitely
with
the
the
spaces that
spaces,
spotted too.
yeah, if you are very sure that your format is correct, check for spaces.
Yeah, watch out for the difference of a tab or a space. A human can't see it, but the computer definitely can.
So we're on our jobs list now. If you've ran any vet jobs before, this would be populated for you. In my case, this is the first vet job I'm running on this browser. So there's only one job in here, which is the one that we've just entered. By default, the page refreshes every 10 seconds, so you don't have to do that manually, but you can if you press on this refresh button. Before we look at the results, I just want to show you a few options. So I mentioned yesterday that if you do any jobs, if you submit any tools jobs on our website, we keep the results for 10 days and then we delete them. But if you want to keep your results, what you can do is you can register an account with us and then save that to your account. And then that way you'll be sure that it won't get deleted. So you can do that in here. You can also edit your ticket, your job. So if you realize that you wanted some more information about your variant and you wanted to add additional configurations, you can do that in here. You can also share your results with your collaborators by generating a URL, or you can delete your job if you don't need it anymore. Okay, so to open up the results for our variant, you can click on Done or View Results. Okay, so at the top, we have our job details. This is just a log of your job, of your web job. Then we've got some summary statistics. So the number of variants that were processed, we only had one variant. So that's one count. Number of variants that were filtered out, we didn't apply any filters, so this is 0. Any novel or existing variants, overlapping genes, overlapping transcripts, overlapping regulatory features. So these two options are not available for Ensembl bacteria, but they are available for our other Ensembl sites. If you scroll down, we've got our results in a tabular format. And in the first column, this is our variant that we processed. I didn't give the variant an ID. So what VEP does is it just gives you the variant in the default format that you submitted. So we've got for the variant, we've got the name, the location. So these are the coordinates. We've got the consequence. So these are the sequence ontology terms. What the effect of the variant is on the gene. If available, we also give you the gene symbol for the particular gene. We've got the gene ID. We've got the protein ID if it's protein coding. And if it's a non-synonymous variant or if it affects any amino acids, we also show you the changes in there. And at the very end, you can also see which feature strand this variant or this result refers to. And there are many more columns that you can show or you can visualize, display. So if you click on Show Hide Columns here in the top left, we can see that there are a few columns that we haven't, that aren't turned on. But I know that we want to find out what gene it affects and what the most severe consequence is. So I just want to make sure that the gene column is turned on and that the impact column is also turned on. So to let us know how severe the the consequences of that variant. Okay. So the first thing you probably noticed when you looked at the results is, why do I have so many results for just one variant? That's a very good question, because a single variant can fall within multiple genes. So what you're seeing here is that this variant actually overlaps all of these different genes. So it overlaps, if we just sort it by symbol, it overlaps 1, 2, 3, 4, 5, 6, 7, 8 different genes in bacteria. So this is why you're seeing multiple results. So we have our list of genes. Now we want to find out what the most severe consequence is. We can sort the table by impact. So just click on the column name, and we can see that we have moderate and then modifier. And if we go back to that table, the sequence ontology table, which I will open on my slide here. It's quite hard to see, but if you look at the last column here, we have our impact. So the highest impact, the most disruptive impact are labeled as high, and then the least disruptive impact are labeled as modifier. So it goes from high, moderate, low, and then modifier. So from that, we go back to our VEP results. We can see that the in-frame deletion is the most severe variant or most severe consequence in our case, because it has an impact of moderate. Okay, so these are the results. So how do you actually download them? You can go just above the results table. There is a download option. You can download your results in the variant calling format, in the VEP default format, or just as a text file. So this would be either a tab or comma separated file, just text that you can then open using Excel or Google Sheets. Okay, so that's all I've wanted to show you for VEP. Are there any questions before we move on to the exercise? All right, so in that case, please go to the training page and open up the Ensemble VEP exercises. I will give you 10 minutes for this, and then we'll come back and continue with the next module. If you have any questions in the meantime, please feel free to add them to the chat or the living document, and George and myself will take care of them. So there's just been a question. There's a question that's come in, and they're asking, how do we look for variants? So in Ensemble, you can't actually look for variants. In Ensemble bacteria, you cannot look for variants. You can only run them through the variant effect predictor. So what you need to do is you need to put your variant into one of the formats that are accepted. So for example, the VEP default format, which is what we used for this demonstration. And then you can run VEP to see what the consequence of that variant is on any overlapping gene. Then there is another question. So these variants, are they just random, or is it something found out from a wet lab analysis? So in your case, it would be from an experiment. But for the demonstration here, this is just a random variant that I made up. But in reality, when you want to find out the consequence of your variant, it would come from some kind of experiment or a paper that you found. I just made this one up just to show you how it would work. Okay, thanks for the questions. Please keep them coming in. And then there's another question. Can we run a random variant in it just for testing? Yeah, if you like, you can. You just need to make sure that you get the reference allele correctly and the coordinate. So for example, I can just show you quickly how how you could run a random variant. I'm going to go to the home page, and I'm just going to use the K12, E. coli K12 genome as an example. And what I want to do is I just want to visualize a random region in the genome. So I clicked on the coordinates, and I'm in the location tab here in the region in detail view. And what I want to do is zoom into some region in the genome close enough so I can see the bases. Just going to zoom in fully. Okay, so once you zoom in enough, you will see the individual bases of your genomic region. So what you can do is you can, for example, just take this one here. This is a T in our reference. So you could just change that to a G and see how that would affect the variant. So how it would look like in the variant call format, sorry, in the default format is like this. Just going to type it into the chat. Let's take that one here. So that's coordinate. So I'm going to focus on this one here to highlight it for you. Okay. So what I've just typed into the chat is a variant that I made up for this particular base here. So you need to write it in this format. It starts with chromosome, then we've got a tab, and then we've got the start position, and then we've got a tab again, and then the end position, tab, reference allele, and alternative allele. So because this is just a substitution, the start and end position is the same number. And for the alleles, the first allele is always the reference allele. So this is the reference allele that we're seeing here. And the second allele is always the alternative allele, so our mutation. So if you want, you can run that through VEP. Just make sure that you select the correct species and see how it would affect the gene. Okay. There's been another question as well. So I found variants by running data in SNP tool, but the same data when run through VEP doesn't show any variant. How do I justify this? So the first thing you could do is just make sure that it's indeed the same bacterial isolate that you've chosen from the species list. You can also check if your variant format is indeed in one of the formats that VEP accepts. And if those don't work, what I would do is share your variants with us, and then we can have a look and see why it's not showing up any results. So if you like, you can maybe share a few variants, and we can run it for you and see what's happening there. What is the purpose of studying these variants? That's a really good question. So in bacteria, mutations can often have a high impact on the species itself. So if you find a mutation in your bacterial genome, it can affect the protein and thus pathways that it's involved in. So studying variants in bacteria can have loads of different applications. So for example, let's say you've just done an experiment, you've introduced a mutation in your bacterial genome, and it's a mutation that allows you to knock out the gene, maybe. So you could, before even starting your experiment, you could try and run, you could try and predict the consequence of your variant and run that variant through VEP. And that's just one of the many applications. So it really depends on what you're studying, what your purpose is for that, for your research project. You're very welcome. Okay, so I'll give you another 5 minutes for the exercises, and then we will move on. Okay, so 5 minutes are up. I will move on now, simply because there are a couple more things I want to go through before we finish. But please keep the questions going. We will answer them in the background. And if you have any questions after today's session, you can always put them into the living document. So I'll keep monitoring it for the next week. So please feel free to add any questions, even tomorrow, if you have any. Okay, so let's continue on. And what I want to talk about now is what comparative genomics analyses we have for species in ensemble bacteria. So it's a very exciting time for comparative genomics. There are a lot of different consortia that are aiming to sequence loads of different organisms. So for example, we've got the Darwin Tree of Life, which aims to sequence all eukaryotes in the UK and Ireland. We also have the Earth Biogenome Project, which is very exciting as well. And the objectives of comparative genomics is to examine the evolutionary relationships and find differences in the genomes or between the genomes. But more importantly, the practical application is what practical application that drives the gene function based on homology. We can learn something about the gene function of a less studied organism or more obscured species by looking at equivalent genes, so homologs, in better studied organisms. We can also learn more about the gene function in, for example, humans by manipulation in model organisms. And all of the animal research is based on this assumption of homology. And comparative genomics is also used to identify any highly conserved regions, which tend to be similar across taxa, and these tend to be highly functional. So in ensemble bacteria, we perform pan-taxonomic comparative analyses. So this is comparative genomics analyses across different taxa. And what we do is we take a number of representative species from each taxon. So we take a few model species from protists, a few from plants, fungi, invertebrate metazoa, vertebrates, and bacteria. And then we perform these comparative analyses on all of these representative species. And you can find any pan-taxonomic comparative analyses data in the Gene tab under pan-taxonomic compara. One type of comparative analysis data that we produce are gene trees. So for this, we take a representative protein of each gene from each species. That's why we also call them protein trees. And then we classify the representative proteins into families or other clustering methods. We align them. We reconcile them against a species tree to produce the gene tree. And then finally, from the gene trees, we can make homolog predictions and potentially infer the function of a gene in a different species. Another comparative analysis data that we produce are homologs. So homologs are genes that are shared by different species and that are descended from the same common sequence in a common ancestor. So you can find two types of homologous relationships in ensemble bacteria, and these are orthologs and paralogs. So on my slide here on the right, we have a simplified phylogenetic tree or phylogeny to explain these relationships. So we've got these terminal nodes. The letter represents the species, and then the number represents a gene in that species. So we've got 3 different species here. We've got this rod-shaped bacterium, this kind of blob, and let's say this is a kind of virally genome bacterium. So these genes, all of these genes here in the terminal nodes, have descended from the same common ancestor over here. So in other words, they all share the same evolutionary origins. So if we look at the two different homologous relationships we have in Ensembl, we've got the orthologs. So orthologs are genes that have emerged through a speciation event. So this is when a population splits into two or more new species. And these speciation events are highlighted in these blue internal nodes in the phylogeny. So focusing on the phylogeny, we can infer that B1 and C1 are orthologs because they have emerged through a speciation event at this common ancestor. As well, B2 and C2 are orthologs of each other because they have emerged from a speciation event from this common ancestor here. So that is a one-to-one ortholog. These are examples of one-to-one orthologs, but we also have orthologs that are described as one-to-many. And this would be an example of S and B1, C1, B2, and C2. So one to many, because these genes have emerged from common ancestor at this node here from a speciation event. The second type of homologous relationship you can find are paralogs. So these are genes that have emerged through a duplication event. So this is when a gene or part of a gene has two identical copies in a species. And the duplication event is highlighted as this red node in our phylogeny on the right. You can have within species paralogs. So an example of this would be B1 and B2. So if we look in our phylogeny, we've got B1 and B2 here. These have emerged through a duplication event at this common ancestor. So these are within species paralogs. Then we've got between species paralogs, and an example of this would be B1 and C2. So B1 up here and C2 down here. So these have emerged from a duplication event at this common ancestor. So to summarize on this slide, homology describes the relationship between genes and the events of how these genes emerge. And in Ensembl, we give you information about orthologs and paralogs. Okay, so the comparative genomics data in Ensembl bacteria, compara just stands for comparative genomics, that we have are all found in the gene-based, in the gene tab. So these are all gene-based resources. And the data that you can find are phylogenetic trees and homologs that have been inferred from these trees. You can also find pairwise protein alignment-based homology, so homologs that have been found by pairwise protein, by aligning proteins to each other. And you can also find information about protein families. Okay, so what we're going to do in this demonstration is we're going to look at the LAC-Z gene in E. coli, strain K12. And we're going to find out what compara data we have. So we're going to look at gene trees and homologs. And this is the same gene that we used as the demonstration yesterday for our gene annotation part of the session. And I'm going to copy the species and the gene name into the chat in case you want to follow along. Oh, there is a question in the chat. So the question is referring to the homology. And I'm just going to read it out loud. Is the C1 and B2 also a paralog between species? C1 and B2 is, yes, correct. So this is C1 and B2 have emerged from a duplication event over here, and they are between species, so two different species. Actually, I'm going to write that down in here as well. That's a very good point. Thank you. Okay, so where was I? Yes, the demonstration. So I'm going to switch to my browser, and I'm going to stay in the E. coli K12 strain, but I'm going to switch to I'm going to change to the species information page. So to go there, you can just click on the species name. And this is the information page for our species. And I'm going to use the search bar in this page to search for LAC-Z. Okay, so we have one result for LAC-Z in E. coli K12. This is a beta galactosidase. We've got the gene ID, we've got our species, our coordinates, and the gene trees that it's part of. So I'm going to click on the gene ID just to go to the gene tab, which is where all the comparative analysis data is found in Ensembl. And just to double check, if you look at the top of the page, we're indeed in the gene tab. On the left-hand side in the panel, we have this section called the pan-taxonomic compara, or pan-taxonomic comparative analyses. And for this particular gene in E. coli, we've got a gene tree, we've got orthologs, and we've got paralog. There's no protein families information for this. So let's just look at the gene tree for now. Okay, so each gene tree has its own Ensembl ID. So that's what you're seeing here. So if you wanted to share, a URL with your collaborators to this gene tree, you can just copy it from here. In our gene tree, we can see the number of genes. We've got 11 genes. We've got 7 speciation nodes, 1 duplication node. So paralogs are paralogs emerge through duplication events, and orthologs emerge through speciation events. We've got 2 ambiguous nodes, so it's not quite sure which event happened here. And then the number of gene split events is 0. Scrolling down, you can see your actual tree here. And there is a notice just above that says that this tree is only part of the super tree. So what you're seeing in this tree down here in this diagram is just a very small version of your actual gene tree. When you see gray triangles like this, that means that the branch has been collapsed to make it a bit easier to look at. So if you want to see the full tree, you can click on the triangle, which will open a pop-up menu. And in the pop-up menu, you can expand or collapse all your branches. So if you look here, you can expand this particular subtree, or you can expand all subtrees. And I'm going to expand all subtrees just to show you how it would look. Okay. Oh, I'm going to expand this one as well. All right. So now we've expanded our tree. Our gene of interest, so the one that we searched for, is colored in red just to highlight it in our tree. So this is LAC-Z in E. coli K12. And we've got homologs in Klebsiella, we've got Enterobacter, Shigella, and so on. On the right-hand side, you can find an alignment of your of these orthologs. So anything that's colored in green are aligned regions, and anything that's empty like this are gaps in the alignment. If you go down to the bottom in the legend, you can see what the different shades of green mean. So if it's empty like this, so if there is a gap, that just means there's a 0 to 33% aligned amino acid sequence. And then we've got 33 to 66 and then 66 to 100. Then we've got our nodes, our nodes colored in the different events as well. So just like in the presentation, we've got the blue representing speciation events. We've got the red representing duplication events. And you can also find out what the different colors for the collapsed nodes mean, but we only have this gray one here. Here you can also, just if you scroll down to the bottom, you can also change your views. So for example, if you only wanted to view paralogs in the gene tree, you can set that here. You can also collapse trees by the taxonomic rank, or you can expand the entire tree as well. You can also download your tree. So there are different options to do so. You can download the image, which will open this pop-up menu, and all you need to do is select the format that you want. Or you can download the data, so the actual alignment or the tree. And, oh, sorry, that was a bit quick. If you want to download the data, so not the image, but the data, you can click on export. Select what type of data you want to get. So you can get the alignments in these different formats, or you can get the tree in these different formats. Underneath, we've got previews of each of the formats. So depending on your downstream analysis, you might want a very particular format for that. And then all you need to do is either download or you can preview it in your browser. Okay, so this is the tree. Next, I want to show you the orthologs. And I'm just waiting for Ensemble to load. Okay, so in the ortholog, you will find a list of your species set. So remember, in the pan-taxonomic compara, we take representative species from each of the taxa. So we've got 12 species from vertebrates, 24 from invertebrate metazoa, 11 from plants, 7 from fungi, 13 from protists, 83 from bacteria, and 24 from archaea. And you can show details for particular species sets only if you like. So by default, we're seeing all of the species. Here on the right, you can also find out the number of orthologs and what type of orthologs they are. So we've got 4 one-to-one orthologs, 4 one-to-many, and then we've got 24 many-to-many orthologs, and 142 species don't have any orthologs. Scrolling down, we have this table here that just gives you more information about each of the orthologs. So if you have a species in mind that you want to compare the LAC-Z gene in E. coli to, you can just search the table for that. So let's maybe look for Klebsiella, because I just remember that from the gene tree. You can type that in, and that will filter your table down. So for Klebsiella, this is a one-to-one ortholog. The ortholog in Klebsiella is this. So the one, the ID that starts with KPN, and the gene name is LACZ as well. We've got the target ID, the query ID, and you can hover over these columns to find a description. We've got the gene order conservation score, whole genome alignment coverage, and the confidence. The GOC score and WGA score coverage are not available for ensemble, so that's why, for ensemble bacteria, so that's why you're seeing an NA. And then the high confidence just tells you how confident we are that this is an ortholog, or how confident our pipeline is about this ortholog. You can view the gene tree. So this would just send you back to that page that we looked at before, but highlighting the Klebsiella in the gene tree. You can also view the sequence alignment. So if you click on view sequence alignment, select what type of alignment you want to view, either the protein or the cDNA. Just going to open up the protein. Just waiting for Ensemble to load. Okay. So this is the protein alignment between LACZ in E. coli K12, which is what we searched for, and its ortholog in Klebsiella. Up here, we just have the details about the alignment, some summary statistics about the alignment, and then underneath we have our actual alignment in ClustalW format. So the first line is our reference, in our case, LACZ in E. coli K12. And then the second line is our ortholog, so the one that's in Klebsiella. And the ClustalW format, what it is it gives you different symbols indicating the alignment between the two sequences. So the star means the amino acid is the same. Then we've got empty, like this one here, so there's a complete change. I actually forgot what the colon means, but I think it's, let me find that out. So if you want to open up a, what's the word? Documentation for this, let's open this up here together. So we can go to help and docs at the top of the page and find comparative genomics here on the left. And what we want is protein tree, actually available alignments. That. No, that's not it. Protein trees. Okay. There used to be a page that tells you what these mean. I know there is that, we have that for Ensemble, so I will share it with you. But you can also Google it. So the symbols are just indicating how well conserved the amino acid residues are between the two species, between the two sequences. Okay, so you can download this alignment. Just click on download homology, and it's similar to the export option in the gene tree, where you can just download the alignment in FASTA, ClustalW, and so on, or you can download it in OrthoXML and PhyloXML formats as well. Okay, so let's go back to our presentation. So now we have more time for exercises. So if you go back to the training page, open up the comparative genomics tab, and continue with the exercises there. In the meantime, I will answer the questions in the chat. I think there's actually one question that I can answer now. And the question is, collapse means the endpoint. So just a reminder what this collapse refers to. If you go to the gene tree, if you go to the gene tree, what you will see is some of these branches are kind of put together, collapsed, to make it easier to look at. So that the tree isn't too big, so that you can navigate the phylogeny a bit better. So this is just done for convenience. But what you can do is you can expand this tree. So here we've got 6 homologs in bacteria. What you can do is you can expand this tree by clicking on the gray triangle. Then expand the subtree. And now your tree has gotten much bigger. So you'll find that the collapsed tree has been expanded to show you the individual branches in there. But what you can also do is you can collapse them back. So all you need to do is click on a node, on an internal node. And collapse this node in the pop-up menu. And that will make your tree a bit smaller again. There you go. I hope that makes sense. Okay, so I'll give you 10 minutes for this exercise, and then we will continue on. Okay, so it's 8:25 Indian Standard Time now, so we're going to continue on. I can see there are a few questions in the living document that I will need to do a bit of research on. So I will get back to you on them today. But in the meantime, please keep the questions coming. They're very, very useful. Okay, so for our final module of our session, I want to talk about custom data. So you can visualize your own data in Ensemble to plot any genomic features on a reference genome. So for example, you might want to map virulence factors or antibiotic resistance genes to a genome. You can also visualize pathogenicity islands of in a particular species, or display RNA-seq data against a reference to visualize the gene expression pattern. In Ensemble, you can either upload your file. There is a 20 MB size limit, but if your file is any bigger than that, you can also attach it as a URL. And there is no size limit for this. But just keep in mind that the bigger your file is, the longer it will take for Ensemble to load this. So while there is no size limit, it will take much longer if your file is any bigger than 20 MB to visualize your data. On this slide, you can find the different file formats that you can upload to Ensemble. So these include feature annotations in gbrowse, GFF or GTF, BigBed, and variants in VCF. You can also visualize continuous values. So for example, any signal data for GC% calculations or ChIP-seq data. These can be uploaded in bedgraph, WIG, and BigWig files formats. And you can also upload sequence alignments in PSL and BAM format. So where do you host any files that are bigger than 20 MB? So usually your institute would have an FTP site where you can upload your data to. But if your institute does not provide this, you can also use either Cyverse or Figshare, which are websites where you can upload your data to generate a URL. Please note that Google Drive links or GitHub links will not work. They have been proven unreliable, so you'll have to use a different way to store your data and generate that URL. But Cyverse and Figshare are pretty good. And that's pretty much it for this session. So I'm going to show you how you can upload, or how you can upload your data to Ensemble. And then we can move on with the exercises where you will do this on your browser. So I'm going to switch to my internet browser. And what you need to do is you need to select your species of interest first. So you need to select your reference species. As an example, I'm just going to use our E. coli K12 genome here. So I'm going to click on the species name. And in the species information page, under genome assembly, you have an option to display your data in Ensemble bacteria. So all you need to do is select or click on this link. And this opens a pop-up menu where you can add a custom track. So this is uploading your data. You can name your track however you like. So for example, if you wanted to visualize any pathogenicity islands in E. coli, call it something descriptive because this is going to be your track name. Then you need to select your species of interest, which is E. coli. And then you can either paste your data in here, upload your data using this button, or if you have a URL, you can also enter your enter your URL into the text box. If you need to enter your data into the text box, Ensemble will ask you to provide what file format it is in. It won't be able to detect it automatically if you paste it in here, but if you upload your file or if you provide a URL, it should be able to see or to find out what file format you've just given it. And then all you need to do is just click on Add Data, and that will save it in the configurations. And then you can go to the location tab. So for example, maybe you have a particular region that you wanted to visualize. You need to switch to the location tab. And then you should find your file, your data that you just uploaded as a track here in the region and detail view. And I want you to try this out yourself. So if you go to the training page, open up the custom data tab, and there's an exercise to add a file. So this is a URL that we're giving you to visualize this this region, and then you can explore what genes are mapping to the region. In this case, this is in the gluconacetabacter genome. Okay, so this might take a bit of time, so I'll give you 10 minutes for this, even though it's just uploading, but there will be a lot of you uploading at the same time, so I'll give you some spare, and then we'll finish up for today. Okay, so it's 1/4 now, and we are getting to the end of our ensemble bacteria workshop. There is still an outstanding question in the living document. I believe George is currently working on that, so thank you very much. But just remember, the living document will be open for another week. So if you have any more questions, please do enter them in there, and we will get back to you. But out of curiosity, I made another Slido. You can join the same Slido that we used yesterday, but if you don't have it open anymore, just go to your browser, either on your phone or your computer, and oh, let's see my mouse. Okay, sorry, I couldn't see my mouse there. So you want to open slido.com in your browser and enter the number 7198778. And what I'm interested in is your species of interest available in ensemble bacteria? So hopefully you had a chance to explore the list of species. And if you did, were you able to find your particular species of interest? In the case of bacteria, did you find your strain of interest even? Okay, a lot of you have found your species. This is great. And I remember from the word cloud yesterday that a lot of you are working with some very popular bacterial species. For those who couldn't find their species at all, I'm very sorry about that. But you don't have to worry, because you can actually request that species to be added to ensemble bacteria. So all you need to do is send us an email, and I will share the email address with you in a minute. Send us an email, include the sequence ID, so the GCA ID, to that particular isolate or strain or species that you're working with. And then we can feed it back to our colleagues and see if we can add it to a future release in Ensemble. Now, just remember that all the data in Ensemble is open access. So you want to make sure that the sequence or the genome assembly has been submitted to a public archive. And these public archives are either ENA, the European Nucleotide Archive, NCBI, or DDBJ. So the sequence has to be submitted to one of these three resources for us to be able to upload it and annotate it to Ensemble. Oh, someone's just asked for the link of the Slido, so let me just share that. Here we go. Okay, so that brings me to the end of our session. So thank you for staying until the very end. And I know it was a lot of information, but please don't worry. The materials will be there for you in case you wanted to come back and try the remaining exercises yourself. So the first thing I'm going to ask you to do is to fill out a feedback survey. And this feedback survey really helps us improve our courses for future participants. I'm going to enter the URL into the chat. Here you go. But you can also find the URL in the training page. So if you still have your training page open, just above the demos and exercises, there is a link for the feedback survey And I'm going to open that just to show you. So there are just a few questions in here. Shouldn't take any longer than a few minutes, and I will give you time for that now. At the end of the survey, you also have an option to receive a certificate of attendance. So if you wish to have a certificate of attendance, just click yes. And in the next page, just enter your email address in there. Okay, I'll give you a few minutes for this, and then we will wrap our session up. Oh, sorry, everyone. I think I shared the wrong feedback survey in the chat. So let me replace it with the actual survey that was from a different workshop. Thanks for pointing that out. The link in the training page should be the correct one, and I've corrected the URL in the chat now, so you should be able to open the correct one now. Thanks. Okay, so while you're finishing up the feedback survey, I just want to go through a couple more slides with you. I will keep the feedback survey open for another week as well. So if you didn't get a chance to fill it out today, please do so by the end of next week. So that's when I will close it. Okay, so what did we learn about ensemble bacteria in these past few days? So we've looked at the ensemble bacteria genome browser, which is genome browsers integrate and allow you to search information about genetic features um, in a genome. And ensemble bacteria specifically maps genes to genome sequences. So these are annotations. And what we do is we perform analyses on these data using tools and comparative genomics, our comparative genomics pipeline. And we also provide any corresponding entries to external databases. Ensemble is what we call an added value resource because it brings together information from a wide range of other databases into a single site. And we've only explored the Ensemble Bacteria website today, but we have other sites as well. So we have the main site dedicated to vertebrate genomes. If you're interested in fungal species, for example, you can find them in Ensemble Genomes, which is dedicated to non-vertebrate species. We've got the Rapid Release website and the Ensemble Beta site, which I mentioned at the beginning of yesterday's session. And we also have archives of our previous releases. So if you're working with an older release, please don't worry. The data is still available to you on our FTP site. You can access Ensemble in different ways. So we've looked at accessing the data through the browser, but if you want to integrate Ensemble data into your pipelines, you can do so via our REST API, Perl API, MySQL servers, and we also have an FTP site if you want to download any whole genome data. If you enjoyed the course, please do recommend us to your colleagues. So we do offer other courses as well. We offer a browser course, which is a full day course. So about 8 hours where we teach you all about the Ensemble browser. We also have a REST API course, a more advanced course on how to access this data via the REST API. And we also offer a Train the Trainer course where we teach you how to teach Ensemble. And if you are interested, please don't hesitate to contact us. So our email address is helpdesk@ensemble.org. Or if you want to find more information about organizing a course with us, you can go to the training page under hosting. There is more information about the timelines, the details on what we can teach, and so on. Okay, so we're also on social media. So we are on Blue Sky, LinkedIn, X, and Facebook. And if you want any news about Ensemble in general, or if you'd like notifications on job opportunities, you can go to our blog at ensemble.info. And then finally, I mentioned that if you wanted more advanced bioinformatics courses, these are available on the EBI training page, and the training materials on there are free as well. All you need to do is just search for your field of interest. So for example, metagenomics, and it will give you a list of recorded webinars and past courses and online courses that you can take on metagenomics. We also have more Ensemble tutorials on our website and our YouTube channel. And if you have any questions at all, our email address is helpdesk@ensemble.org. So if you have if you found that your species of interest is not in Ensemble, just email us at helpdesk, and we'll see what we can do about it. And if you want to stay up to date as well, you can join our mailing list here. Then finally, sorry, we're a couple minutes over, but if you're using Ensemble data, please don't forget to cite us. So the most recent paper for Ensemble Genomes is the 2022 paper. So just make sure you cite that in your research. And then, of course, it's not just George and me in the team. We're a team of about 100 people in Ensemble, and it takes a huge team effort to keep the resource going. So thank you all very much for staying until the very end. I hope you learned loads about Ensemble bacteria, and I hope
मैडम
you get to
बताइयो
use it for research.
so thanks Loy for your sessions, and thank George. And on behalf of Decode Life, I would like to express our sincere gratitude to you for delivering insightful and engaging workshop. The sessions was highly informative and well received, providing practical insights that will greatly benefit students in their work. We truly appreciate the time and effort your team invested in making this workshop a success. We look forward to future collaborations and hope to host you again soon. Thank you. Thank you all. Thank you all the
Wonderful.
participants.
Thank you so much. Thank you. It's been great.
And this brings about the end of this microbial genomics workshop. Thanks all the participants. Thanks Ensemble EMBL team. And we believe that our participant enjoyed past month of 1 month of learning with us. Thank you all. So with these words, I seek permission of Louis and George for ending today's session.
That's fine with us. Thank you.
Thank you all. Best wishes.
See you soon. Bye.